All right, let's start. Good morning, afternoon, and evening. Hola, hello, bonjour, hola. Ni hao, konnichiwa, 안녕하세요, and hello all. Welcome and thank you, SynBYSS people, for joining us the 187th seminar, and I call it a Technical University of Denmark Day. I just came back to San Diego after my 17-day nonstop trip to attend two conferences organized by Professor Jennifer Doudna at Har and give 6 talks at 6 different places, including KAIST, Seoul National University, and Pomona, California, where Microbiome Editing Golden Research Conference was held. It was exciting to meet and discuss with so many old-time and new colleagues and trainees, although I slept for only 3 to 5 hours a day over the entire trip period of 17 days. Okay, it is my tremendous honor to introduce very briefly our pioneer speaker, Professor Dina Petranovic. She is the senior executive advisor at Technical University of Denmark and also was the CSO in the past. I met her for the first time at J. Craig Venter Institute, where a global forum was held, and she was a representative of Denmark with the Professor Jens Nielsen at the time. She kindly accepted my invitation to this webinar, and she has a fabulous career going different direction. And then I believe she's gonna talk about wonderful, journey she has took until now. Dina, thank you so much for your contribution to the scientific society and also your leadership in global biotechnology, and the virtual podium is all yours now, and thank you again.
Thank you so much, Taesuk, for inviting me, and hello everyone from dark and cold Copenhagen, Denmark. It's a pleasure to be with you and tell you a little bit about what I have been doing over my 25 plus years in biotechnology. So I'm Croatian of origin. I was born into a family of scientists. My mom was a chemist, my dad was an MD, and both of them did research in molecular genetics. So they met in the lab. They ran their research groups at the institute, and they were teaching at university. And so I sort of, I don't know, maybe I didn't have a choice but to become a scientist. But when I was a child, I actually wanted to be an archaeologist, and I assume it is, growing up in the late 70s and 80s, influenced by Indiana Jones. So, I thought I was going to be an archaeologist all the way until I was 14. And then around that time, I was lucky to go to CERN in Geneva, where we had friends at the time. And CERN was also famous back then. And they were building one of the first detectors, particle detectors. And I had a chance to go underground and visit all of that. And I was like, okay, I'm going to be a physicist. And for a few years, I really wanted to study quantum physics. And then things changed when I was in high school, and I had a really fantastic teacher in molecular biology. I had very nice education, both in Croatia and in Paris, where I lived with my parents. And so this teacher really inspired me. So it's not so much my parents, even though they were geneticists and I was in the lab all the time, but she really inspired me for molecular biology. And I think it was because first time it was somebody external besides a family member talking about molecular biology. And you know how it is, if it is your mom and dad, then it's maybe not as cool. And it's much more cool if it is somebody external. And so I was like, okay, this feels like this is the right fit for me. And so I studied molecular biology. Then I did a PhD in molecular microbiology in Paris. And then I did a postdoc in Denmark for several years. Then I was professor in Sweden for 10 years. In the meantime, I visited a few labs in US, and I will come back to that. So I was at Harvard very briefly, then 2 times at MIT, and also at Caltech, at Francis Arnold's lab, who was your first guest, I understand, on this series. And so I think very early on, I realized that what I really like about science is the discovery, but I don't really care about who is doing it. So I did not have this feeling that it has to be me doing it for, glory and name and so on. Sometimes, many scientists that we know are really driven by, personal accomplishments. And I realized very early on that, I love the stuff that we discover, but it doesn't have to be me. So I get really excited about great discovery that somebody else made. And, if we get a great paper, I realized I'm more happy for my student, my postdoc or my PhD student than I am particularly for myself. And I think that was a very good discovery for me, because I realized that I will never be able to discover everything that I want, but what I can do is support many people along the way. And so that kind of triggered these thoughts about how do I get as much things done as possible, even if it cannot be done by my own hands or the hands of the group that I'm personally running. And so I started to get involved with all kinds of things in leadership at university in the hope of kind of being able to influence bigger things to happen. One of the things that I did, I was for 6 years CEO of Molecular Frontiers, which is an organization that is located in Stockholm, and it's run from the Royal Swedish Academy of Science. It was established by the chair of the Nobel Committee back in time, and he had this vision of bringing molecular sciences closer to public and to children and young people, but general audience. And so I became CEO of that. And for 6 years, we went around the world and organized events and, did online activities in order to reach out audiences and talk about molecular sciences. So not only biology, but also chemistry and physics and so on. And I thought this was really inspiring. And it was really kind of like an additional fuel to my day job, my regular job as a group leader and a scientist. And that, I think, kind of put me on this track on thinking, well, I can do this, right? I can have a standard job, the stuff that you see normally, and then to have something else on the side that, if your motivation or your energy level drops down for one type of work, you pick up with another type of work, right? Maybe you don't need actual rest. Maybe you just need to step back from something you are getting, hammered into. And so I really loved that, and I did that for a long time. And then apparently I have too much time on my hands. My husband is always teasing me about that, because then I decided to enroll into a business school and do an MBA. So for 2 years, I went back to school, and I wanted to study something that I don't know anything about. And at the time, I was like, business. I know nothing about business. I have no idea how the real world works, so I'm going to study that. And so I did that for 2 years, and I have a master's in business and then an executive MBA. And that was at the same time when I was doing some innovation. So I took some of my own research and did some, small projects related to innovation to see how it could become potentially relevant for spinning out, or how to handle my own intellectual property and so on. And that was really useful. And actually, one of the awards that I'm sort of proud of is to getting an award for IP in Sweden for something that didn't work. And I thought that was really great. But I think it was really important to realize what works, what doesn't, why it doesn't work. I had a mentor from a very big pharma company who was an advisor for that. So it was such a nice exercise to be able to, take science out of the lab and see what can work. Luckily, other things that I have done later on with my science did lead into useful innovation that is licensed to some other startups. So I'm not running my own company, but I'm collaborating with these companies that are using my IP. That leads me also to another thing that I'm doing now, which is I'm chair of the board of a small startup that somebody else has set up. But also it's one of these things that I get really excited about what other people are doing. So again, it is not necessarily my own science that needs to be something that excites me. I'm very happy to put my time and effort and networks and connections to help other people achieve their goals. One thing that I have learned along the way of doing these kind of things has been the importance of networks and collaborations with colleagues within my own immediate research environment and also broader than that. So I have reached out and actually talked and collaborated with people from academia, industry, diplomacy, regulators, funders, and so on. And I think this is really a little bit special how I constructed my career compared to many other people. I worked in the foundation, the Novo Nordisk Foundation, which is one of the wealthiest foundations in the world that funds life science research. I was senior manager there for biotechnology. Then I became chief science officer and chief partnerships officer of one of their big research institutes here in Denmark for 5 years. And now, from January this year, I left the institute to basically be the strategic advisor to the leadership of the university. So I'm part of the leadership team and helping the university to do all kinds of science. So I don't only focus on biotechnology, even though it's my, of course, primary focus because of my main competencies there. But I like having the big picture of what we can do at university at strategic level. But I think my advice is that if you're not super attached that the science has to be yours personally and that it has to have your name as the inventor and author and so on, maybe you have so much knowledge and insight and creativity and ideas that you could actually contribute to other people's, efforts and creativity. And it can be somebody else's startup, and it can be somebody else's institute, and it can be somebody else's university or outreach organization. And doing a lot of things has actually helped me figure out what I'm good at and how I can contribute to this very broad perspective of what biotech can do, and use it hopefully for the betterment of all of us. So yeah, I would say when you're young, it's good to be focused and become specialist in something. But as you are growing more and more experienced, think about how you can use that experience to actually build up the entire ecosystem and the entire environment for the benefits of more people than just yourself.
Fantastic. Oh my God, this is the one of the best talk I ever listened to. So thank you so much. Also, you and I have very interesting common thing. Actually, my high school teacher also made me, chemist first, and then another, teacher in college also made me molecular biologist later on. And then my journey also not linear at all, non-linear. And then I absolutely agree with you regarding bigger impact. Because the reason I start this webinar is I wanted to be scooped by young people because I want to solve the 10 global problem, and I realized I cannot solve any of them. If that is the case, but I will still wanted to solve all the 10 problem, the only way I can solve that problem is helping other people, encourage other people to scoop me, and then I will solve all 10 problem, and then I don't need to solve problem myself. So
Yeah.
that's my conclusion, and then that's why I started this webinar series. And then you are absolutely right. I lovely you mentioned one thing I wanted to do in the future. So thank you so much.
Thank you.
Yeah. All right. Now, the main speaker of today with a longer introduction. Dr. Michael Grome is a postdoctoral associate at Yale University's Systems Biology Institute. There, he conducts research in synthetic biology, genomic recording, and also engineering of biomaterials in Isaac lab. His work leveraged genomically recorded microbes to address challenges in translational decoding, protein engineering, and biocontainment of GMOs. And that is actually all I very interested in as well. Michael earned his PhD at Yale, pioneering DNA nanotechnology approaches for membrane remodeling. Beyond the lab, Michael is a passionate science communicator and educator known for his outreach in scientific thinking and biotechnology. He regularly mentors students, creates educational programs, and also speaks at scientific and public events such as TEDx and Yale's Pathways to Science on how science and biology intersect with society. Such an outreach activity is an indication of leadership, and I believe he will become a great leader in near future. Michael, thank you so much for your time today. Please take it away. all yours now. Thank you again.
Thank you very much. Let me share my screen and right here. Yes, I want to say thank you very much. I'm deeply honored for this opportunity. And I will say timing is wonderful because I have a nice 1-year-old at home and a wife who are just as sick as I am with the nice RSV going around. So my voice is very hoarse. So I hope that you can bear with me. But I would like to say a major take home from this for especially grad students at home. Is that if I can do this talk this sick, you have no excuse to not give lab meeting. So I hope we can get that. So from here, I'm going to start. Here's a basic presentation outline that I have. I'll start with background, my personally, then go into some genetic code expansion and recoding background to tell you what that is, go into the construction of a one-stop codon, which was my primary postdoc work, and a little bit about looking forward. And I will say with the background, I went to a public school in upstate New York, and I did first generation in academia. I had no idea of how to get into it or where to go. But ultimately, I was able to find my way through a circuitous path and found a direction that I'm very excited to move forward into, hopefully in an assistant professor position soon. So to start, I did my undergrad at Cornell University, where I focused on plant science and natural resources, predominantly for sustainable agriculture and biotechnology. And I got to do a wonderful internship with one of the co-inventors of the gene gun, Ted Klein at DuPont Pioneer. But from there, I moved to Yale University, where my predominant focus was on exploiting the programmability and plasticity found in nature to, in a sense, construct like synthetic cells. And so in my graduate work, I worked with Dr. Chen-Chong Lin doing Yale Nanobiology Institute, where I worked to kind of build bottom up in an in vitro sense to build toward programmable biomimetic cells. And that went from DNA synthesis to the self-assembly of hundreds of oligonucleotides, of oligos, into DNA origami nanostructures that could, in a higher level, self-assemble and recapitulate natural membrane deforming complexes in pursuit of those biomimetic cells. And so I had some publications if you're interested. Here you can see on the left, you can see natural protein complexes wrapped around in vitro lipid tubules and our DNA versions wrapped around lipid tubules as well, showing that we can mimic size and structure to recapitulate function. From there, I moved for my postdoc to Farron Isaac's lab in Yale Systems Biology Institute. And so this went from in vitro to in vivo, doing top-down engineering. And here we took E. coli to do system-wide genome engineering, translation machinery engineering, and ultimately all to create and expand the genetic code with synthetic chemistries. And so I'm going to predominantly focus on this work today. So here in the Isaac's lab, we focus on biologically inspired engineering. This was my PI's words. He loves words like that. And this research was, it's predominantly Farron Isaac's lab with close collaboration from Jesse Reinhardt. And so this is our recent lab photo. Hopefully it's nice little grins in there. And what we work on is we make precision and multiplex gene editing tools. We do pathway discovery and engineering, biomolecule prospecting and engineering, genetic code expansion to create these synthetic proteins, and biological safeguards with genetic isolation and biocontainment, and many of these associated with my work with the construction of genomically recoded organisms. And what that means, we're going to go over. So going from my background, let's go into background of what genetic code expansion is. So here's your standard genetic code. You have 64 codons that effectively code for 20 amino acids with discrete chemistries and a stop function. And so here you have your amino acid charged with an aminoacyl tRNA synthetase onto a tRNA, and then carried by EFTU elongation factors to the ribosome, where this process continues until a release factor recognizes a stop codon and releases your full protein. Now, beyond the 20 canonical amino acids, there are actually hundreds of what we call non-standard amino acids that have new, diverse, discrete chemistries, many not found in nature, that endow proteins with new properties. And so here's an example of a variety of different side chains with their functions. They can vary in all these different properties, charge, polarity, redox potentials, so that you can really modify proteins and really finely tune a lot of these different properties to give it new functions or enhance existing functions. And so this could be like recapitulating organism-specific post-translational modifications outside of their hosts, enabling light and ligand activation in enzymes and transcription factors for regulation. It could be enhancing or producing chelation and metal activation in proteins, as well as bioorthogonal conjugations within and adjacent to in vivo systems. And we can do this, incorporate them into proteins through what are called orthogonal translation systems. We can have plasmid expression of orthogonal in engineered synthetases, tRNAs, and occasionally elongation factors that when fed or synthesized a non-standard amino acid can be charged and delivered to the ribosome to site-specifically incorporate synthetic amino acids at target codons. And this has a whole host of downstream applications from engineering and protein production. However, within our natural systems, there are still native translation factors that compete with our synthetic ones for those target codons because all these codons have their own functions. And this can result in reduced incorporation efficiencies and reduced purity that leads to either misincorporations with native amino acids or truncated products when it competes with release factors. And so we decided how could we address this? Well, early researchers looked at diversity within nature of genetic codes and decoding. Within the standard code, you can have the same codons read by different tRNAs in different ways. But there are a whole host of alternative genetic codes that reassign codons to different or new amino acids. And these can be single codons or entire codon blocks. And so this raises the question, how malleable is genetic decoding? And can we exploit this plasticity to alter or reassign codon functions and to mitigate that native codon competition? Well, this brings us to genomic recoding. And so looking at our standard code, we actually see that multiple codons can code for the same function. And this is what we call the degeneracy of the code. And so if we take a look at the stop codon block, here you have 3 codons that are all decoded by release factors, protein release factors, RF1 or RF2. And you'll see both of these, we have 2 release factors and they both target the same codon. Well, within biology and engineering, redundancy equals opportunity for change. And so we ask the question, do we need 3 stop codons? Can we free up one of those codons and reassign it to a synthetic function? And do we need 2 release factors? You know, if we have one, if we get rid of one of these codons, do we need both of them reading the same one? Can one pick up the pace? And so ultimately this led to prior work associated with my PI, Farron Isaacs, in which he helped construct the first genomically recoded organism, or GRO. Mouth is dry, still sick. So this involved 3 primary steps. The first is codon compression. They identified the rarest codon, TAG, in the E. coli genome that has about 300 instances. And what they did was they targeted and mutagenized, converted all TAG codons to TAA throughout the genome, eliminating the TAG codon from the genome entirely. Once that was completed, they wanted to liberate the codon to free it from native translation recognition, to get rid of that codon competition. And once the TAG was gone, then you had 2 release factors reading TAA. Did we need that? And nothing reads TAG except for this. So basically, they deleted it. And by doing that, RF2 was able to pick up the pace, and TAG could then be codon reassigned by expressing an orthogonal translation system, a tRNA that targets that codon and reassigns it for a synthetic amino acid, increasing that accuracy of multi-site incorporation up to 95%, yet seen in the field today. And that was back in 2013. And that was really exciting. And the downstream applications were numerous and still going, in which we could now do photo-cross-linkable peptide hydrogels. We could recapitulate human kinase networks that need specific post-translational modifications at specific sites in E. coli. We could functionalize and peptides and increase their half-life for therapeutics with like lipid binding. We could create electrically conductive nanowires using gold nanoparticles. And a work that I worked on, which I loved, was creating biocontainment mechanisms by modifying essential proteins, replacing codons with our new ones, and then making cells dependent on synthetic amino acids for survival. You add the synthetic amino acid, doesn't occur in nature, the cell lives, you take it away, the cell dies. So if anyone's seen Jurassic Park, they had what was called the lysine contingency, where the dinosaurs were auxotrophic for lysine and they needed it to survive. Well, that was dumb because lysine exists in nature, but these are synthetic amino acids, so the dinosaurs won't escape this time. You know, famous last words, right? So that was the GRO. But once you makes these new proteins, but once you edit the whole genome like this, it creates a totally different genetic language. And that creates innate genetic isolation. Viruses and genes that require TAG termination to translate go into the cell and they can't properly translate. So they don't. And then genes that get out that contain your TAG as a sense codon serve as stop. And so out in other systems, they terminate and truncate your protein. So innate isolation. All of these were very exciting. But when we created this recoding system, we had further questions that we wanted to ask. Because, hey, we freed up one codon, but there are 64 codons in the genome. So looking at the standard code, we asked the question, how far can we compress an entire translation function? Can we make codons functionally degenerate and take all these different codons, split them apart for individual functions? And so specifically looking at the stop codon block, we asked, can we compress the entire stop function into a single codon, leaving two unassigned codons that can then be reassigned for dual and specific incorporation of synthetic amino acids that could have enable broader host defense, multiplex functionalities, bioorthogonal conjugations in vivo with different non-standards, and recapitulate complex multiple PTMs to produce therapeutics and human compounds within these E. coli systems. And so to do that, we followed a similar construction pathway of codon compression, liberation, and reassignment. But looking at the genome, you know, there were 300 TAGs. Well, there are 1,200 TGAs. That is 4 times the number of codons. That's a lot more work. So we did it, but, spoiler alert, but we'll tell you how we did it. So ultimately, we identified all instances of TGA within the genome of all these genes and converted them to TAA, eliminating TGA from the entire genome. And we did that through basically designing 1,200 DNA oligos that enable not only individual G to A conversions, but also addressed overlapping genes, about 300 of them, to separate them apart. And our mutagenic oligos basically work by acting as artificial Okazaki fragments that when we knock out mismatch repair proteins or suppress them, we can then incorporate into the replication forks and introduce our mutations. And we can do about 10 at a time within a strain. Well, the original work was we took our first recoded E. coli missing the TAG codon and targeted all the essential genes within 2 clones of this strain. And then once we did that, we were then able to employ conjugal genomic assembly to then reassemble those converted genes into a single essentials recoded genome. and we performed RNA-seq to see how is transcription ultimately modified from that system. And when the cell looked pretty darn good, we then took that strain, split it into 8 clones, and targeted 1/8 of the genome, all the genes within each of those eighths. And then through MAGE, combined with hierarchical conjugal genome assembly, we were able to assemble them into a final fully recoded strain, trying to address background mutations and auxotrophies and fitness issues throughout the whole process. So it was iterative mutagenesis, gene combination, and then selection to build our final strain. And this led to about 1,200 TGA codons converted. We used 16 major deletions using the Blattner group's minimal E. coli genome work as a reference. And this acquired about 800 background mutations because we knocked out repair pathways. But the truth is, we don't know how many of those do nothing, are detrimental, or are essential for the strain to survive, because we are really doing a lot of these conversions. And the TGA codon doesn't just work as a stop codon, but it also works in cellular stress. So there's other underlying things with overlapping sequences and whatnot. So there's lots to learn about. But once we do this codon compression, we then move to codon liberation. And what we wanted to do was we wanted to eliminate the competition from RF2 at the TGA, at the UGA gene or codon. But how can we do that? We can't just delete the release factor because something has to terminate UAA, and RF2 serves essential ribosomal functions. So what do? We engineer RF2 to be specific to the UAA codon and attenuate that recognition. Well, how do we do that? Well, actually, there are a variety of different mutations we had to make to the RF2 structure that you see over here on the left. You know, the release factors have a lot of very interesting biology. They switch and change conformation. They have autoregulation. They have interactions with the ribosome. They do decoding. Too much to get into, but what we'll focus on is down here what we call the codon recognition loop. And this is a loop that is largely associated with decoding and identifying the stop codons in the ribosome. And so looking at that, there's this site, serine 205. And you can see up in the structure that it largely is associated with kind of structuring this codon loop. And there is a paper from the 2000s that showed in vitro, if they converted serine to the RF1 mimic proline, they could actually attenuate UGA recognition and termination without targeting or modifying the UAA termination. That's ideal. That's what we want. And so we made this conversion, which you see kind of destabilizes this loop, and the cell died. It didn't survive, right? So what I did was I identified through comparative analyses of release factors, RF2s, from genomes that had a low number of TGA codons, but a high concentration of release factors. The idea is if there's a lot of protein to terminate a small number of codons, they're probably bad at it. And so identifying only 9 mutations, there are actually 2 of them that worked and 1 we moved forward with. And this is an E170K charge flip. This is on what I call like the charge shield of the protein. And it was actually identified in previous mutagenesis studies that looked at improving recoded strains, interestingly enough. And what this does is in previous studies looking at it from the same group, yes, mutations at this site, charge flips, can alter decoding by the position of the release factor in the ribosome. But this site was shown to not significantly alter codon recognition. And so making this change, we were able to make a viable strain with this S205P mutation. And so we think that it might give it a positive charge to increase interaction with the ribosome, kind of like almost mimicking an increased concentration and frequency. But it might also increase the residence time in the ribosome to allow the protein to kind of recognize and decode that codon. So we wanted to say, okay, does this actually terminate the way we think it does? And so we made this M-Cherry codon YFP read-through assay. And here the X in the middle corresponds to one of the 3 stop codons. And so the idea is that if your release factor terminates at the codon, you will get M-Cherry expression and no read-through to YFP. if you have a suppressor that targets the codon like we do for UGA, you would expect read-through of mCherry and co-expression with YFP. However, if neither a suppressor or a release factor recognizes the codon, you'll get ribosomal stalling and no expression of either protein. And so when we performed this assay, what we found was with the both release factors, you see termination at the UAA codon. You see no recognition with suppressors or with a UGA suppressor or release factors at UAG. But at the UGA codon, when we add a suppressor, what we find is that you outcompete the suppressors with wild type release factor 2, but when you make the mutant release factor, the suppressor outcompetes the release factor. And that suggests that the mutations attenuated UGA recognition and termination in release factor 2. That was exciting. That's what we wanted. Oh my gosh, we can move forward because we didn't know exactly if this would work or not, right? But sometimes it does. And so, but what we did though was we performed mass spec on our constructs. And what we found was that we had a degree of misincorporations from native amino acids, a suppression at the UGA codon still. And the primary one was tryptophan. And that is actually not a surprise because the UGG codon for tryptophan is the fourth codon within the stop codon block. And if you look at the structure of tRNA tryptophan, it has a post-transcriptional modification at position A37, which enables UGA wobble recognition. And so in times of stress, the tRNA trip can outcompete release factor 2 and suppress the UGA codon to extend proteins and alter global transcription. So you have, in order to free up this UGA codon, we have to actually address this recognition of tRNA tryptophan as well and abolish that recognition. And fortunately, through comparison with other organisms, in yeast, there's a tRNA cysteine with a similar modification at the same site that enables cysteine wobble recognition at UGA. But when you convert A37 to G, you abolish that post-transcriptional modification and UGA recognition. So we made the same mutation ourselves and ultimately in the mass spec abolished all instances of tryptophan incorporation, which was super exciting. We didn't know if it would work that well and the cell would survive, but we eliminated dependence from TGA and we can. This is fully enabled by our recoded strain alone. That's the exciting part. And so that rendered the tryptophan tRNA UGG specific. And so looking at our GRO again that we're constructing, and we look at the stop codon block, what you see is this translational crosstalk at the UGA codon. And so what happens is when we tend to think about codons, we think, oh, here's a codon, here's its function. But in reality, within cells, you can have whole hierarchies of tRNAs and release factors that compete to recognize that codon. So whatever is the canonical function might only be the dominant function. And so when you do genomic recoding and you remove these translation factors, you have to keep that in mind. You remove RF2, you get TRP. You remove TRP, you might get cysteine, and so on. So keep that in mind as we disentangle this crosstalk. And so to do that, we had to modify RF2 and modify tryptophan. Only then could we then reassign those codons with synthetic amino acids and create a fully non-degenerate codon block in which each codon eliminates that translational crosstalk and makes functional exclusivity. And so that was really kind of a key component of this work that was exciting. And this could be used to create dual incorporation. So how did we do that? Well, we did a dual incorporation orthogonal translation system. Here we have our reporter system with different GFPs and our dual tRNA synthetases and tRNAs to target each codon. And so to kind of make this relevant for synthetic biology, the amino acids we chose was PACF for UGA, and this is associated with bioorthogonal oxime click chemistry for functionalization. And we use Bach lysine, which is often used in therapeutics, where there's a protective Bach group on a lysine that can be deprotected and then functionalized for later use. And then these were incorporated 3 codons each to make 6 total within an elastin-like polypeptide which is used in synthetic biology for enhanced protein purification, drug delivery, tissue scaffolding, and rare earth metal recovery. Woo, trying to talk. And so to look at our data, we have lots of data in the publication, but to really focus this, we'll look at our dual incorporation reporter that has TGA and TAG, and we'll focus on the addition of both amino acids fed to the cells. And what you find is the difference with the cell between codon compression and without compression, you see a modest increase in fluorescence. It's significant, but not much. But where you really see in the incorporation rate go up is when you create the mutant release factor. Now you have this significant jump in incorporation efficiency by mitigating that codon recognition by the release factors at the UGA codon. But what you see is when we make the mutant tryptophan, you see a drop in fluorescence. And that's actually not surprising because when we did our MS data, our mass spec, what we found was that with the wild type tryptophan, you see about 7% misincorporation from additional amino acids. However, when we make mutant tryptophan, the bulk of that goes away, giving us over 99% incorporation efficiency and accuracy, which is the highest to date. And so that was super exciting. And so from there, the key takeaways from this work is that the TGA codon can be removed from the genome and the cell can survive, maybe with compensatory mutations. The removal of TGA eliminates that dependency and enables attenuation of native recognition from RF2 and tRNA tryptophan, compressing the stop function and enabling reassignment. We have TF hierarchies, translation factors that all compete for codons and intertwine, and translation engineering allowed us to liberate that UGA codon without removing those essential transcription or translation factors. And ultimately, it was recoding plus translation engineering that allowed us to do this highly precise incorporation of each amino acid at their own respective codons without crosstalk. And that was the key component of this work. And so that was my predominant postdoc work. It took a long time. It's a lot of work, a lot of moving parts, a lot of people. But looking forward, we have a lot of exciting downstream applications that I don't have time to go over today, but I'll briefly go into some insights. You see, because when we do this work, there are still some limitations. This is our first go at our genomically recoded E. coli. And so some of the limitations open room for further enhancement and insight. And that includes non-standard protein yields, gross cellular growth, and genetic orthogonality. And so what happens is here we had our highly accurate incorporation efficiencies, right, in our ultimate product. And these produce yields that way outcompete kind of natural non-recoded strains compared to like BL21. However, the ultimate protein production, was only about 20% of wild type protein production without non-standards. So the question is, can we bring this up? Can we make optimizations to translation and OTSs in order to enhance this production? That's an area we're working on. Another one is cellular growth. In the background, we had over 1,500 changes from the original wild type E. coli, but we also had genomic deletions and this 800 background mutations. We don't know what's good. We don't know what's bad. Can we target those and improve fitness? When we look at the growth, you'll see in orange is doubling time, lower is better, and black is cellular density, higher is better. And as we made changes throughout time, you see the cell gets slower and the density goes down. Well, while these still relatively compare decently well with many commercial strains and with other recoded strains, we do have to ask the question, can we further enhance the doubling time, making the cell faster and grow with higher densities, right? Can we target those mutations and translation? And then finally, genetic orthogonality. What we see is we performed phage assays to test, can phage proteins that require, for example, with lambda, essential TAG termination or mu, essential TGA termination, can these infect and translate in our cells? And what happens is what we find out is like the first recoded strains, whenever a strain has release factor 1, it can translate and terminate TAG, making the cells resistant to lambda phage. But without it, the cells are susceptible. Sorry, I said that backwards. With the release factor 1, the cells will terminate and be infected by cells. And without release factor 1, the cells are resistant, just like our ochre strain. However, all of the cells we tested are still susceptible to mu. And the reason, even our ochre strain, where it maintains the original resistance to lambda at TAG, but susceptible at TGA. And the reason for that is what we did was we engineered release factor 2 to be less competitive, to be outcompeted by by an orthogonal tRNA. But given the opportunity without a suppressor, there can still be some degree of termination. It's not abolished. However, we had some variants that did show zero termination and resistance to phage, which suggests that there is room to improve, but these variants were non-viable. So can we enhance that orthogonality? And so this leads us to our areas where we're working on now. So we want to enhance genetic orthogonality of these strains, optimize non-standard protein yields, improve cellular fitness, investigate revealed biology, look into our data and our publication. There's so many wacky biological outcomes that come from these strains with mutations and whatnot. We have students working on biomaterial applications and looking forward to applying this to new recoded genomes. And so fortunately, we have some publications that we're working on are about to come out, hopefully shortly, that address many of these early cases that I'm really deeply excited about, especially enhancing our properties and investigating that revealed biology. So I want to thank you all for listening to my talk. Questions, comments, job opportunities. I'm on the market, right? And this was really a combined effort. I got to help lead a team on this, and it was a phenomenal experience. And thank you all for listening and bearing with me.
Fantastic, fantastic. I really love your enthusiasm, and then you are really great communicator. It's fantastic. Let me ask Dina whether she has a question or comment.
I was thinking, so you said about revealed biology and so on. I guess it's not necessarily the easiest thing, even in E. coli, to reveal the biology when you're going to have so many additional mutations that are just arising from the fact that you are, removing a codon and so on. What is your approach to reveal biology, as you call it? I mean, I assume it's some approach for systems biology / modeling / something, but I would like to know what you have in mind, and if you can hint at least what kind of biology are you revealing?
Oh, excellent question. I'm excited you said that because this is some of my favorite parts. When we make all of these changes, we really do stir up and kind of in some cases break some pathways. And so whenever we want to look at kind of weird, wacky biological outcomes, we always have to try to compare it to wild type E. coli strains. And so like two examples. Okay, maybe I'll stick to one, but one phenomenal example is we found that certain TGA codons, when we converted, led to immediate changes in phenotype. Just a single nucleotide switch from a TGA to TAA altered global transcription. Mm-hmm. And in many cases, end biological phenotype. And so what happens is what we can do is we can look at those codons. And there's this one case where we're like, okay, maybe this is associated with a suppression event. So we would mutagenize this codon with all different codons. And what we found was one specific codon showed up. And it was one that was never identified in E. coli to suppress and change transcription in times of stress. And so what we do is we try to modify that in the wild type E. coli as well and see if we have similar phenotypes. And so that's one example where it's really exciting, where it's like we may have just revealed a biological mechanism that nobody's been able to see. And that's largely because it was just stirred up in this massive engineering effort.
That sounds really fun. We had a paper published in Science some years ago where we discovered that one single mutation, single nucleotide mutation, contributed 80% of the phenotype of survival of yeast under higher temperature. So it basically was a mutation in an enzyme that synthesizes sterols, and it switched yeast from having ergosterol in the membrane to having fecosterol, which is what archaea has. And so it changed yeast growth at higher temperatures, which is just temperature also for industrial production and so on and so on. So sometimes, yeah, really wacky stuff comes out of of course, there were plenty of other mutations, right? But we could eliminate them eventually and zero in on this one enzyme, one amino acid change due to one nucleotide, changing the entire pathway for sterol synthesis, membrane rigidity, survival at higher temperatures, et cetera, et cetera. Super
Well,
weird things. So I think you're on the right track of finding weird, cool stuff that is potentially also going to be useful for some applications and so on.
well, that's fascinating because then it shows just how easy sometimes it can be for an organism to transition to different environments as well. Is
Yeah.
sometimes it just needs that context and that one opportunity.
Yeah,
That's
nobody knew that yeast could make fecosterol and become an archaea.
So Dina, do you have any other question or comment?
Well, we could talk about many things, but if anybody else has any questions, I'll let younger people jump in.
Sure. Let me kind of ask the similar question that kind of always kind of govern my entire career. So the question is a little bit philosophical in a sense. So you kind of show amazingly complex engineering over the years. This is the concept is simple, but the real execution is so complex. But the same thing was done by nature over the years by evolution. The question I have is, I could keep asking what is the governing principle in biology that kind of applicable to any biological system, but I still kind of try to find some of them. Of course, evolution is one of them. But the question I have is, I believe the chemistry somehow many billions years ago kind of determined this three stop codon should be stop codon rather than other codon. But you probably cannot answer the question why. So if we go back to the billion years ago, start the evolution again, do you think three stop codon gonna be the same stop codon again in the new era? Or the different question is why we have boring 20 amino acid rather than 21 or 22, rather or only 19, because 21 might be having the different world, different nature, different creature, whatever, more complexity, or 19 might be simpler. I don't know. This is kind of hunting me my entire my career in biology.
Oh, don't be honored by it. It's exciting. So actually, that's a good question because there's a lot of directions to go within the evolutionary context,
Sure.
right? Sometimes you develop a protein, and something we're finding out with release factors that I hope to release soon, which is exciting, is that sometimes you get the development of highly conserved systems. Once it starts, it's locked into place, and the combinations of chemical changes needed to change that is just too great to overcome. One change won't get you there, two won't get you there. You need five. But the possibility of all those five happening in the right condition is just too great. Mm-hmm. However, what we see with the shift, there's a lot of recent work in alternative genetic codes and their evolution. And a lot of it seems to suggest that there's cases where you get shift not necessarily from the mutations in the genome so much, but in the shifts of competition between tRNAs and release factors that can alter which codons are assigned to which amino acid or function based on stoichiometries, sometimes governed by the environment or whatnot. And that can shift which mutations are available to then mutate and change genomes later. And so you'll see shifts where codons can be reassigned in this way. But you say, oh, genomes that use 20 amino acids, we have context-specific incorporations, right? Where there's mechanisms for selenocysteine or pyrrolysine. We have post-translational modifications that do that as well to expand protein chemistry. But there's a recent paper that came out that should be of interest where it suggests that a genome might have reassigned most, if not all, codons. I believe it's the UAG codon to an alternative amino acid, pyrrolysine. Now, there might be more work to look into to what degree that's a complete refinement, but it kind of shows that given context and given the evolutionary pathway, once a chemistry is established, you think of like a tech tree, especially for you gamers or whatever, of what available pathways are now available from that point.
I see. That's very
Yeah.
interesting. So the simple question in that case, I mean, so I love the movie called Matrix. Probably you
Yeah.
are
Yeah.
too young to remember that one, or
Oh,
you know that the movie. Okay, so that means, I mean, if you start the world again or Matrix again, seventh time or eighth time, you believe we may have same
oh,
system with a 20 amino acids, three stop codon, or you also believe in alien because they start with a different initial condition and end up having 24 amino acid or some different amino acid set
oh.
and a completely different nucleotide set.
Yeah, I think it could be that way. You could imagine, let's say we started with the same building blocks on another planet, where let's just say it was colder, or let's say it had more, I don't know, UV light or some different chemical composition there, right? Or even if it just started again and maybe the wrong wave crashed down,
Mhm,
I don't know. It could be the case that maybe there are many different chemical possibilities or arrangements in which life could have taken.
mhm,
And maybe in early life, there were multiple competing configurations, but one of them won. And once it got into that configuration, there's almost little chance or none at all of going backwards. And so that started off. So if we started again, maybe we would see differences, maybe different bases. There's researchers working on alternative nucleotides, different amino acids, what compounds are available and what quantity to those life forms as they produce. But also, if you just look at the standard code we have, there's some suggestion with simulation and crystal data that maybe the UGA codon behaves slightly different in the ribosome than UAG or UAA,
mhm,
just in terms of like base orientation.
mhm.
And so maybe different tRNAs, different modifications that could shift that, maybe different proteins. The release factor in eukaryotes, the nucleus, has almost no homology with the release factors in prokaryotes, and they might have been convergent evolution.
Mhm,
And so if you have a different protein, maybe you could have different stop codons,
mhm,
you
mhm,
know?
mhm.
So and it's just that, oh, we already had these amino acids, so another protein was able to move in.
I
So there are
see.
really so many possibilities.
Absolutely. So, that I was thinking about that question because if life is the constant moment of choice, and then if I choose something different, I may just working with the Dina at DTU rather than working in United States, or I might be doing singing in front of the audience rather than doing seminar. So this is all about the life and also biology. So that's why I'm asking that question. So
Thank
absolutely
you.
wonderful talk. I love it. I see, now, 11:04, and then I believe this is a dinner time for dinner. So we need to close. Uh, so let me close and then we, probably continue to chat if you want. Okay, so thank you all for joining and staying today. And we will meet again on February 5, Thursday, the same time, the same Zoom link. We'll have the Professor Jens Nielsen, the CEO at Bio Innovation Institute, also in Denmark, and a National Academy member of all three categories, that is science, engineering, and medicine, and also Dr. Simon Du Olesnitz at Harvard Medical School. As usual, the follow-up informal chat will occur without recording. Please stay here if you are interested in chatting with us. I'll promote you to panelists who can speak and show your handsome and pretty faces if you wish. And thank you again. I stop recording. Just 1 second.